In this video, I will briefly review the nucleic acids. Nucleic acid monomers are made up of three different components. First of all, we have the phosphate group. This is a functional group containing a phosphorus atom as well as 4 oxygens. Depending on the conditions in which the nucleotide is existing, there may or may not be hydrogens associated with some of these oxygens. The second component of an individual nucleotide, or a monomer of the nucleic acids, is this sugar molecule right here. Using organic chemistry notation, this is the first, second, third, fourth, and fifth carbon. So this is important because the fifth carbon, or 5' carbon, we can use this notation to essentially talk about the beginning of the molecule. Essentially, all enzymes read nucleic acids from the 5' end down in this direction towards the 3' carbon here. And again, just one more note about that. The prime, this apostrophe means prime. So just a notation on the carbons. Okay. The third component of nucleotides, in addition to the phosphate, the sugar, and this here, the nitrogenous base. Okay, this base contains nitrogen. We can see that there are several different types of nitrogenous bases, and each one contains a name. Now, this name is essentially what is given to the entirety of the nucleotide, even though thymine itself refers specifically to this base, this particular grouping of atoms. And so there are 4 that are present within DNA: thymine, guanine, cytosine, and adenine. Now we can see that thymine and adenine are joined together by these dotted lines, which signify hydrogen bonds. Now, thymine and adenine are called complementary bases, or complementary nitrogenous bases, because they are always attracted to one another. Similarly, guanine and cytosine are attracted to one another via these hydrogen bonds as well. There are 3 between guanine and cytosine, 2 between thymine and adenine. Okay. Attached to the nitrogenous bases, which are complementary to thymine and guanine, we can see yet again a sugar and a phosphate. A sugar and a phosphate. However, it might look a little bit different than over here, and the reason for that is that these are essentially upside down. This is the 5' end, and this is the 3' end, as opposed to 5' up here and 3' down here. What this is showing us is the fact that strands of DNA, right, so this grouping, right, this chain of nucleotides, are what are called antiparallel. This strand over here goes from 5' to 3'. Its complementary pair chain goes from 5' to 3'. Okay, so again, this is called antiparallel. Okay. Now, there are a lot of different ways that DNA can be portrayed, and you'll see a lot of these in different textbooks and online sources, so let me just walk you through them briefly. If we move down, we can see that the nitrogenous bases have been simplified to just signify or just say a letter. Okay, so A for adenine, T for thymine. Again, we can see that there are 2 hydrogen bonds joining together these complementary bases. Okay. We can also see that the phosphate group has been simplified just as a P with a circle around it. Once again, we can still see an individual nucleotide: phosphate, sugar, nitrogenous base. Phosphate, sugar, nitrogenous base. Of course, they have been joined together by a specialized bond called a phosphodiester bond. Okay. If we move down a little bit farther, we can see yet another type of notation. Here, even the carbon backbone, that is the phosphate and sugar, phosphate, sugar, has been simplified to just this little rectangle. Okay. Ultimately, our genetic code is stored in the order of these nitrogenous bases. So the most important thing to portray when looking at DNA is, of course, what is the identity of the nitrogenous base. Okay. Once again, A is complementary to T, G is complementary to C. Okay. To even further simplify the portrayal of DNA, what we can see here is, again, this carbon backbone, right? Each of these is implying the sugar and the phosphate, and the nitrogenous bases are linked together in between, forming this very recognizable double helix structure. Okay, so again, I just wanted to show you a few different ways of portraying the same molecular structure, the same molecule. Okay. Now, DNA is stored in chromosomes. We have 23 pairs of them in the human genome, and they range from about 50 million base pairs, which is essentially this pair of nitrogenous bases. Right? So essentially, base pairs means nucleotides, or pairs of nucleotides. So again, the chromosomes range from about 50 million of these pairs to about 250 million in a single chromosome. Surely, those molecules are really big and clunky to move around. Also, they contain all of the genetic information for how to code lots of different types of proteins. And so, in order to actually read a particular gene and actually make a protein, make other types of molecules, we use a transcript. So, we don't actually use the DNA itself out in the cytoplasm. So I'd like to briefly talk about the relationship between DNA and the transcript molecule RNA. Okay, so what we can see here is, the DNA is super long, millions of base pairs long. If this particular region here is a gene coding for a particular protein, we want to make that protein. And so instead of opening up the entire chromosome, what we do is we locate the gene itself, and there is another video that explains this this signaling process. But essentially, at the beginning of the gene, there's called a promoter region, and that is going to signal an enzyme called RNA polymerase to attach to the specific region of DNA and start to do something that's called transcribing a copy of this gene. Now you can think of the entire chromosome as essentially like the cookbook. We don't want the entire cookbook. We just want one particular recipe. And so the RNA polymerase is going to create a polymer of RNA. It's an enzyme, thus ACE, going to make a copy of just one particular recipe. So one particular gene, not the entire cookbook. Okay, so what we can see here is that RNA polymerase is going to kind of fold itself around the DNA. And from the 5' to 3' direction, it is going to attach complementary nucleotides. Okay, so here we can see G goes with C, G goes with C, T goes with A. But wait, what about this? Okay, U, I have not talked about that before. U for uracil is only present within the RNA and not within the DNA. On the other hand, T for thymine is only present in the DNA and not in the RNA. So here T is complementary to A, and if we were making an mRNA copy, A is complementary to U. And that's the only difference as far as RNA versus DNA is concerned in that context. Okay, so again, the RNA polymerase is going to kind of slide along the DNA gene, and it is going to catalyze reactions, forming bonds between complementary nucleotides, that is complementary to the DNA gene. And so what we end up with is in mRNA what is called transcript or copy of just the gene. Okay, the gene is in the DNA, it's part of the entire cookbook. We don't want the whole cookbook, we just want the recipe for chocolate chip cookies, and so that is what this is going to allow us to do. If we look closely, we can see U is complementary to A, T with A, U, G and C, again, and A and U. Okay. Now, mRNA, a type of RNA, is yet another type of nucleic acid. Okay, if we come up here, what we can see is that DNA and RNA really do have a lot in common. Both, again, are nucleic acids. They are built of multiple nucleotides, but the nucleotides themselves are a little bit different, as is the molecule. First of all, deoxyribonucleic acid versus ribonucleic acid, RNA versus DNA. Deoxy implies that we're missing an oxygen, and that's absolutely the case. All right, so first let's take a look at an RNA nucleotide. Phosphate would be here, nitrogenous base is here, and on the 2' carbon in the sugar molecule, we can see an OH, okay, oxygen. Now if we come over here to the DNA nucleotides, we can see that the 2' carbon on the sugar has nothing. Okay, so actually this implies a hydrogen, but no oxygen. So the 2' carbon has been deoxygenated, right? No oxygen, compared to ribonucleic acid, which has this oxygen. Okay. Second main difference is that DNA forms this double helix. There are 2 strands of carbon backbones, each with these complementary nitrogenous bases. And of course, the hydrogen bonds between the bases actually holds the molecule together. Okay. RNA, on the other hand, is only formed into a single strand. Yes, RNA is capable of kind of folding on itself and forming a 3-dimensional structure due to the formation of hydrogen bonds, but in general, it is just a single strand of genetic code. Finally, RNA, as I said before, contains uracil as a nitrogenous base, which is what this one is right here. DNA, on the other hand, only contains thymine. Okay, so essentially, if you are transcribing a gene from the DNA into RNA, instead of writing a T, you would simply write a U. Thank you very much for watching this video on the nucleic acids.